This is a short talk for my poster presentation, ChromMiniGraph, a space-efficient minimizer-based pangenome reference graph and haplotype mapping tool. So to start, let's talk about the motivations of ChromMiniGraph. So no single genome can represent a diversity in the human population, and using a single reference genome creates reference biases. So the goal of ChromMiniGraph is to allow the creation of pangenome graph that is compact but readable, relatively fast to create, and able to map reads efficiently and accurately. So the way we achieve those goals is, for compact but readable, we choose to use minimizers, which, as a reminder, is the smallest k-mer in a sliding window of W. And for it to be relatively fast to create, we use an algorithm known as colinear chaining and an efficient range minimum query data structure. And we also wanted to be able to map reads efficiently, and that is achieved by using banded dynamic programming and topological linearization. So I just want to briefly talk about what CMG is and it's not, and what it does and doesn't do. So CMG is different than a minimizer de Bruijn graph because it does not collab identical k-mers. And it stores individual k-mers with subsample reference positions instead of unitic paths. So in that sense, it's different than some other graph genome tools, say MiniGraph. And CMG creates a pangenome reference, maps read to the pangenome reference, and assigns haplotypes to reads. It performs base level alignment for the best haplotype and output a BAM file. And it provides tools to convert the graph to GFA format for visualization. But unfortunately, as of right now, it does not map read to a reference graph generated using other tools, and it does not compress the reference sequence. So the main algorithm underlying the entire ChromMiniGraph software is the colinear chaining algorithm. So the goal of colinear chaining is to collect a chain of maximal local alignment anchors so that the order of selected pairs satisfies the order in which they appear in both the reference and the read, such that the collected chain covers the maximum area of the reference. So in this case, T is our reference and R would be our read. So colinear chaining is proven to be a very reliable method for estimating near optimal global alignment using pairs of local alignments. So in this example, we can see this is a valid and optimal chain that is highlighted in the bold edges, right? So we have a collection of local matches, right? So collection of local alignments. And so we're trying to find an optimal global or near optimal global alignment using this set of anchors, that's what we call it. So the graph is constructed through an iterative reference to graph chaining, and at each iteration we chain a new reference sequence onto the current graph and merge k-mer nodes to create multicolored nodes if necessary. Reference to graph chaining uses colinear chaining on a linearized coordinate, and it can be solved efficiently using range minimum or maximum query. So this is a graphical representation of what we're doing in the iterative reference to graph chaining. So we start with a reference graph. So at the beginning, it's just going to be this one chain, but as we slowly build our graph, it's going to be this slightly more complicated graph with possibly complex topology. And we can add nodes and merge the nodes onto the chain. And in this step, we first linearize the graph to obtain a linear coordinate of our ChromMiniGraph, and then we do colinear chaining on the linear coordinate instead of the possibly complicated graph coordinate. As to read mapping, it is done using banded dynamic programming similar to other software like Minimap2, and uses topological linearized coordinate for the gap function instead of the more costly DAG chaining or partial order alignment. We also check for color consistency during chaining. And below here is our objective function that we're trying to maximize. And as you can see, it's a dynamic programming equation that is recurrence. So here A and B are just scaling factors that can be arbitrary, and it's optimized later and chosen to be an optimal value. And a few things to note here. So here we are using L, which is our linearization function. That basically says we're using a linear coordinate instead of the graph coordinate. And by ensuring if the anchors are not compatible, we use infinity. That ensures the path coherence of the chain. So the goal of topological linearization is to obtain a linear coordinate for each node on the graph that best approximates the graph topology. Ideally, the set coordinate should optimize for the following two properties. One is known as spatiality, and the other one is known as monotonicity. Right? So it's all very intuitive notions. So spatiality is basically says nucleotides and k-mers that are physically close together within a genome should have similar coordinates, or let's say linear coordinates that are close to each other. And monotonicity says the graph genome coordinates of successive nucleotide or k-mers within a genome should be monotonically increasing. Right, so those are very intuitive concepts that shouldn't need too much explanation. So there are classical graph algorithms for generating a topological linearization, which is literally just the topological sort algorithm. So DFS, as we can see, it finishes all vertices on the current branch before exploring the other branch. On the other hand, BFS, or as we call it, the Kant's algorithm, explores each branch before moving to the next vertex. So in conclusion, BFS is more desirable because it interleaves vertices to prevent a large insertion. Right? So as we can see here, we have a large insertion EF after our node CD between D and G, and this is something that we don't want. Topological linearization, however, fails to provide a good estimate of the graph topology around regions with unbalanced superbubbles. So this is an example of unbalanced superbubble. So it has a very short branch over here and a very long branch up there. So to fix this issue, we first use a linear time bubble identifier, or superbubble detection algorithm, to identify unbalanced superbubbles and then score the chain using the sampled reference position at neighboring vertices. And this is just an example of why linearization doesn't work as well across a superbubble like this. So here are some preliminary results of our software. So we use ChromMiniGraph to construct graphs for human chromosomes 11 and 20, and each using 16 different reference genomes. The serialized file is a binary file that can be read directly by ChromMiniGraph for fast read mapping. The tool also creates a much smaller simplified graph, which can be used for visualization. And a fully indexed graph file takes about the same storage space as the starting references, but then it can support efficient read mapping. So in a sense, this gives us a graph self-index, right? So here's the benchmark results. As you can see, our starting reference is 1.1 and 2.3 gigs, and the resulting graph file is only 1.2 and 2.3. And when I should also note that for the simplified file, I didn't put the number up here, but it's on my poster. For the simplified graph, it's much smaller. For chromosome 20, it reduces the size to 29 megabytes, and for chromosome 11, it reduces the graph file size to 60 megabytes. And we can also choose to store the index only, so without storing the reference sequence, right? So you can actually choose whether or not you want to align, you want a base level alignment for the mapping step. It is on by default, but you can turn it off, and in which case only creates a graph index without storing the reference genome explicitly. In that case, it also greatly reduces the file of the index. We also ran some tests on simulated data. So we simulated 10 sets of references of different lengths, each of 10 haplotypes. And then we generated long reads with read depth 25 that approximates a long read generated by nanopore sequencing. We evaluate the accuracy and quality of the mapping, and the average accuracy turns out to be consistently above 90%. So that means 90% of the reads are assigned to a correct and unique haplotype, unambiguously. So here is the graph of our simulated test. As you can see, the mapping time is still slower than other software, or let's say the state-of-art software like MiniGraph, but the memory consumption is actually better than other softwares like MiniGraph when we don't do an explicit base level alignment. So in conclusion, ChromMiniGraph is a proof of concept software for a novel minimizer-based self-indexed pangenome reference graph, and it uses topological linearization and colinear chaining instead of the more traditional DAG chaining to improve efficiency. And we employ a linear time bubble detection algorithm and subsample absolute reference position to improve chaining performance around large and complex structural variations, which may be present in our reference genomes. And preliminary test shows promising results on both actual and simulated data, and although more optimization are still needed to match the state-of-the-art pangenome graph builders like MiniGraph. The next step, we are going to hopefully add support for generating graph for more than one chromosome. So it is one of the limitations of the software right now, that you have to create one separate file for each chromosome. But ideally, we want to be able to, say, have a large reference file for the entire human genome and create one graph for each chromosome, but have those graphs stored in the same file as connected components. And we also want to tune parameters in the read to graph chaining objective functions. That's the A' and B' in the previous slides. And also, of course, we want to improve multi-threading and add tooling to convert graphs generated by other software like MiniGraph or VG to our ChromMiniGraph format. Thank you for listening. And again, my name is Jun. You can contact me at the email below, and please check out my poster. There should be a link on the page that you access this video, and I'd love to talk to you guys in person during the poster sessions. Thank you.